All right, so today I want to finish up with the idea that I didn't finish up with last time. That was this homunculus. Then I'll try to do some eye anatomy. I kind of edited what I was going to talk about last night, so you won't get quite my full lecture on the eye, just to try to fit it in. But we'll do eye today, ear on Wednesday, and then we'll start cardio and move in to the cardiovascular lectures for midterms. All right, so last time I was telling you about this guy here, Wilder Penfield. You can maybe, some of you who are interested in reading more like historical texts from medicine should put him in, particularly those who are interested in neuroscience at all, should put that one on your reading list, maybe your like 15-year reading list or something when you have a chance. Also Osler and Cushing, those guys wrote a bunch of stuff that the one he talked to, he studied with, that I talked about last time. What Penfield did could be reduced to this idea of the homunculus, and so here is actually a model of it that he apparently had made, and this is simply representing the distorted features of this character are representing the spatial allocations in the sensory cortex of the parietal lobe that service the various parts of the body. So if you were to look at this, this means that there is a much larger place in the sensory cortex of the cerebrum devoted to hands, lips, tongue, and much less real estate devoted to your back and your the less sensitive areas of the body. And that's because I remember when early on we talked about receptive field, the size of area that a sensory neuron services on your skin. The receptive fields are very small on the hands, which means you have a very high density of sensory neurons, So the mapping of all those sensory neurons in the cortex is going to take a much larger proportional area of the cortex. Pretty straightforward concept, I think. So here's how it works. Here is the parietal lobe, and a gyrus is one of these ridges, and a sulcus is one of the crevices, the valleys in between. And there are these sulci can be a little bit variable, but there is one that is pretty universally observable, and that's this central sulcus that runs right down here. And it defines a pre-central gyrus and a post-central gyrus. That post-central gyrus is the primary somatosensory cortex. This is the part of your brain that indeed has a little map of a human body on it that corresponds with the sensory receptors on the skin. So if you were to take a slice through that post-central gyrus, you would see essentially this. So this is looking at a more or less kind of an oblique frontal or coronal section of the brain, and you can see how you can see the mapping of the various portions of the body. The hand has got a large region, and the back and arm and buttock and leg has about the same amount of area devoted to it as the hand and fingertip. So and that's a reflection of the sensory neuron density. So again, when we were talking about sensory pathways, I talked about the first order neuron, which is the actual sensory neuron that may be in your finger, the beginning of which is in your finger or whatever. The cell body living in the dorsal root ganglia, and then ascending to some point and having a synapse in the spinal cord, decussing, and second-order neuron decussing and going up to the thalamus, where it's going to synapse on the third-order neuron, which will then project to the primary somatosensory cortex in that post-central gyrus. So what Penfield was able to do was stick a little electrical stimulator at a little electrode at maybe the brain tissue. He mapped this area out by sticking an electrode in and having the patient say, oh, my hand is tingling. I feel it right back here. Or my fingertip is tingling. I feel it right here. He was able to, in fine detail, map out this somatosensory mapping. All right, well, on the other hand, if we look just next door, is in the precentral gyrus, we have what's called the primary motor cortex. And analogously to the primary sensory sensory cortex, we have this mapping of all of the efferent neurons that are going to control the various motor, the various skeletal muscles in the body. So there is a functional map of the body very similar to that in the somatosensory cortex that lives just on the other side of the central sulcus. And likewise, we have fine motor control in our hands for picking things up and doing whatever, and a lot of fine motor control over our tongue and lips as is required for speech. However, we don't have the same kind of fine motor control over the muscles of our buttocks or something like that, for example. And so the corresponding area in the motor cortex that correlates with the hand or the tongue is much larger than you would find for the muscles of the torso or otherwise. This does not mean that the muscles of the leg or the torso are smaller than the muscles of the hand. It just means that a single neuron is going to control many more, the motor units in the gluteus or the bicep or the flank, any of these muscles, the motor units are much larger. So a single neuron is going to control many more muscle fibers, whereas in the hand and tongue, the motor units are much smaller. So a single neuron is only going to control a few muscle fibers. All right, and thus, the point of origin in the motor cortex for the hands is going to be much larger because it takes many more neurons to control the muscles that are related to the use of the hands. Does this make sense? So the area in the primary motor cortex is directly proportional to the density of motor units. All right, there's a high density of motor units in the hand. Those motor units are very small, and so they get a much more area in the primary motor cortex. All right, so this is going to lead us into the last slide, which is looking at the somatic motor pathways that are descending from the primary motor cortex. And the sensory pathway, I talked about there being I talked about there being three, right? First, second, and third order neurons. And in the motor pathways, there's only two. There's the upper motor neuron, and then there's the lower motor neuron. And there's really two types of paths. There are motor neurons. All of the upper motor neurons, their cell body is somewhere in the precentral gyrus in the primary motor cortex, and these are general somatic efferent fibers. They're going to descend through the brain and synapse on a lower motor neuron. They can maybe turn up the lower may stimulate the lower motor neuron, or they could also inhibit it, depending upon the actual needs of the of whatever action is trying to be taken. So they take two different paths. These lower motor neurons can be of two types. They can pass out of the central nervous system via a cranial nerve. And if they just pass out of the body via a cranial nerve, they are innervating an effector that is on the ipsilateral side to the upper motor neuron. All right, lower motor neuron is innervating an effector on the ipsilateral side to the upper motor neuron, meaning both on the same side. All right, or they can be exiting the central nervous system through a spinal nerve. A spinal nerve. If they're going out through a spinal nerve, the upper motor neuron is going to decussate before synapsing on the lower motor neuron. Also a general somatic efferent. And the cell body is going to be in the gray matter and anterior nuclei in the gray matter of the spinal cord. And the axon then extends out and goes to the effector. Now, what's interesting about this is that if somebody has a stroke, for example, I should show the next slide, but I hit it. If somebody has a stroke, for example, by looking at the pattern, like say they have a stroke somewhere in the parietal lobe along the primary motor cortex, or anywhere along this pathway, you can essentially tell where that stroke is just by looking at the pattern of muscle paresis or weakness that they're having, right? Are they having a face drop and a contralateral paresis in the hand, for example, or are the signs localized to the same side of the body? That's going to tell you where the lesion is, because the upper motor neurons of cranial nerves versus skeletal nerves have a different decussation pattern. All right, I'm going to stop it there, though, or else I risk not being able to talk about the eye at all. Are there any questions about that concept? All right. So let's skip into the eye. I cannot, maybe out of my own whimsical vanity, skip my my haiku. Behold such a sight. Angels sing in their design, eye and ear sublime. All right. These lectures are about the eye and the ear. Some of my favorite, because I find that both hearing and sight to be profoundly mysterious aspects of human experience. So fun to talk about. In a lot of the little historical blurbs that I give, I admit to being, if not Eurocentric, at least focused on the Mediterranean and Mediterranean cultures, but I do not want to fool you into thinking that that's the whole story, or even necessarily the beginning of the story, or the biggest, most awe-inspiring parts of the story. This guy, Sushruta, from what is today India, was a remarkable individual. So he lived about 3,000 years ago, and he was in the city of Kashi, and he was one of the originators of the Ayurvedic tradition in medicine. So we could talk a lot about this guy. He was also one of the first people. He was the first person to write an account of refined sugar. He was truly a Renaissance person 2,500 years before that had any meaning. But he, in his Vedic texts, he described, amongst the host of different diseases that he described 76 ocular diseases, which is pretty amazing to me. It gives me goosebumps thinking about 3,000 years ago. And he was able to treat 51 of these surgically. He was the first person to, by the accounts, successfully remove a cataract. He was the first cataract surgeon, which is a clouding of the lens. So he was able to do that surgically, and this is before aseptic technique, and the person was able to heal. So yeah, Shruta, maybe it'll pique your curiosity, and you'll learn more about him. An incredible individual. Let's come forward a long time, a couple millennia, to this guy, John Freak, who certainly had some refined taste in hairstyles. He was the beginning of the modern age of ophthalmic medicine. And his studies were centered here at Mooresfield in London, England. If you ever get to visit London and you're interested in ophthalmology at all, it is worth a visit. I won't say much more just to move on here to the actual material. All right, so the first way to think about the eye, we're going to have to go through a little bit of anatomy here. There's actually a huge amount of anatomy in the eye, and I'll just touch on the important parts. So the main purpose of my lecture today is to hit photoreception. I want to talk about photoreception, how that actually works. But we'll get a little bit of the anatomy in here. So the eye can be broken down into three layers. The outer layer is called the fibrous layer, and it's made of the cornea, which just means the clear fibrous layer. It means thin cornea, clear. Pardon me. And then sclera. Sclera means white. Cornea is clear and sclera is white, so the sclera is the white of your eye. These are strong fibrous layers that do contribute to the focusing power of the eye. Each of them, we'll talk about that. The cornea being clear, in addition to the lens, also adds a little bit of optical refractivity to light. When people get RK or radiokeratotomy or whatever, the LASIK. Has anyone ever had that? You guys are too young for that. But it's better to wait until you're a little bit older. Make sure your eye is fully done changing shape. But yeah, the cornea is what gets engraved when they do that. It gets inscribed. And the sclera is this fibrous framework that gives the eye its rigidity. And we'll talk about the significance of that when we talk about accommodation in a moment. The next layer of the eye is the vascular layer, and we call the layer as a whole the uvea. It has a few different parts. So in this diagram, it's labeled in brown. There's the iris of the eye. That's the colored part of your eye that has the pupil that dilates or constricts. There's some muscles in that part of the eye, in the iris. Then there's the ciliary body. It's another ring of muscle that's a little bit deep to the iris. The ciliary body is made of the ciliary process and the ciliary muscle. We'll talk about what that does in a bit, but specifically, or in general, it helps change the shape of the lens so that you're able to accommodate on near versus far. It changes the shape and the focusing power of the lens. And then going deeper into the structure of the eye, we have the rest of the uvea is called choroid. And choroid, in the same meaning as we used it when we were talking about the choroid plexus in the brain. All right, so the choroid in the brain did what? What did the choroid do? You guys remember? It, let me see if I can give you a clue without telling it to you. It was the opposite of the arachnoid granula. How about that? You guys remember any of that? The choroid in the brain made cerebrospinal fluid. It was what produced cerebrospinal fluid in the ventricles. And the choroid in the eye produces the aqueous humor, which is the fluid that fills the eye in the anterior cavity. So you see that anterior cavity is behind the lens, or I'm sorry, in front of the lens, but behind the cornea. The anterior cavity is actually broken up into the anterior chamber and the posterior chamber. Posterior chamber being posterior to the iris, anterior chamber being anterior to the iris. And then there is a posterior cavity, which itself is filled with the vitreous humor. Vitreous humor is this jelly ball. Haley said that in high school she got to dissect an eyeball. Has anybody else ever cut apart an eyeball? Yeah. So there's like, it's kind of like, I don't know, that like when you were a little kid in like grade school and you go to a Halloween party and there's like a haunted house and they have a bowl full of grapes that some mother has lovingly peeled. It is kind of like that. It's this gelatinous jelly mass in the inside of your eye. That's the vitreous humor. All right. Anyways, choroid is where it is the vascular layer. It makes this fluid that fills the inside of the eye, and it also carries all the blood supply to the eye. All right. And then lastly is the neural layer. The neural layer has two layers to it. The superficial one, I guess, thinking about it from the outside of the eye in, is the pigmented epithelium. It's just contains melanin, etc. And then the neural part that has the photoreceptors and the accompanying nerves that will transmit, that do photoreception and transmit sight into the eyeball. All right, so here's a little bit of more detailed anatomy. Here's the anterior chamber of the anterior cavity, the iris, the posterior chamber of the anterior cavity lens. Light comes in. The visual axis, the light will hit, depending on the angle that it comes in, it'll hit anywhere in the retina. But the center of your field of view, the thing that your intention seems to be drawn to the middle of, that light that is forming the center of your field of view falls on what's called the fovea. The fovea. Fovea just means a depression, a small indentation. And what else is new here? There's a bunch of more anatomy that I've either covered or don't think is important to our discussion. So I'm going to move on. Well, I should say this. So there's a lot of times people think the optic disc is the center of view. That it is not. This is actually your blind spot. We'll see what that means in a moment. But it's the fovea that is your center of view. All right. So accommodation. Accommodation is. So I'm going to go through a whole list of ophthalmic terminology here, and accommodation is the first one. So accommodation is simply your eye's ability to change the shape of the lens to focus on something near versus something far. When you look at something far away, that near thing, which is in the foreground, is blurry. However, when you look at the thing that is near, the thing in the background is blurry. And this simply is because the eye, under the control of the autonomic nervous system, is changing the shape of the eye. So this is how it works. For close vision, let's actually start with distant vision. Distant vision. When you're looking at something distant, you need a longer, flattened, stretched out lens. Okay? For that to happen, this muscle that surrounds it in a ring around the ciliary body, the ciliary muscle is relaxed. And being relaxed, it allows this fibrous tunic, the sclera, to pull on the lens. It's attached by these suspensory ligaments of the lens. And that sclera it's like a spring. It sort of opens up a little bit in a circle. And these ligaments pull on the lens and flatten it out. This is for distance vision. And then if you want to look at something near, that muscle contracts, kind of like a drawstring, pulling a sac closed, all right, the orifice of a sac closed. And in so doing, it relieves the tension on the suspensory ligaments, relieves tension on the lens, and the lens is able to go back to its resting shape, even though the muscles are contracted, which is a little bit thicker, all right? So when you're looking far away, ciliary muscles are relaxed, lens is under tension. When you're looking at something near, ciliary muscles are contracted, lens has the tension released and becomes fatter. You guys getting that back and forth relationship? All right, next term is astigmatism. Is anybody astigmatic in this classroom? Few of you, yeah. So what happens when you go around without your glasses on for a while? When you first found out you were astigmatic before they got you corrective lenses, did you have headaches? This is often the case of astigmatic people. So astigmatism simply means that the shape of the cornea from one side to the other is out of whack somehow. One of them is a little bit shaped a little bit differently, so that the images that are coming into the retina are from one side is distorted compared to the other side. And the brain is having a really hard time, the cortex is having a really hard time like squeezing one image and mapping it and synchronizing it with the image on the other side. Often leads to headaches. So when you look, these people will look at things, sometimes an image can be highly distorted. Obviously that's not what that looks like, that's not what it's supposed to look like, or they can get a little bit of double vision. This is in contrast to emetropia. Emetropia just is normal vision. So this big dial you see over here, if you are astigmatic and you close one of your eyes and you look at that dial with your astigmatic eye, at the distance between each of those bars should seem, to a normal person, to an emetropic person, they should be uniform. But if you are astigmatic, the distance between the bars should be a little bit distorted, perhaps. Maybe between 9 and 10 is wider, and between 6 and 5 it's narrower, something like that. Okay. Next terminology is visual acuity. This simply means the clarity of vision. All right. So a person who is nearsighted, that is a person who's able to focus on something near away, but the distance is blurry, is called myopic. And this is because with an uncorrected lens, the lens is, the focal point of the light is in front of the retina. The focal point of the light is in front of the retina, such that the actual image that they're focusing on is a little bit blurry. It's a little bit blurry. To correct this, we put a divergent lens on. And then a person who is hyperopic or far-sighted, the focal point, it doesn't match. So like the shape of the lens doesn't match with the length of the eye perfectly, and the focal point is thrown behind the retina, all All right, so it hasn't fully focused by the time the light receives, by the time the light arrives at the retina, and we can fix this with what's called a converging lens. A converging lens helps bring that focal point forward so that the image is in clear focus when it hits the retina. So as people get older, almost everybody becomes hyperopic. They lose the ability to focus on things that are near to them. That can start, well, you can have it your whole life. Yes?
I mean, if you're
No. Yeah, myopic. It can a little bit. It can a little bit, but these two things are so presbyopia or hyperopia due to age is not related to the shape of the eye. So myopia and classic myopia and canonical hyperopia is related to the shape of the eyeball, that depth of the field in the eye. Whereas presbyopia, the problem with presbyopia is, remember that the lens needs to change shape to focus on something near versus something that is far. And as you get older, the lens is just not as elastic as it used to be, and giving rise to this hyperopia of old age. And we call this presbyopia because, like, a presbyter at a church is like an elder at a church, and so they call it presbyopia, meaning it is hyperopia of the elderly. All right, yeah, that was a good question. The difference in canonical hyperopia is the shape of the eye, and whereas presbyopia is the inflexibility of the lens. All right, so on to the retina. The retina, so within the field of ophthalmology, it seems like that's a subspecialty, but itself has subspecialties. There are ophthalmologists that can be retina docs. There can be that specifically work on problems of the retina. There are neuro-ophthalmologists who spend their time looking at the optic nerve, which you see right here. So when we look at, this is a picture taken with what's called a fundoscope. The fundus is basically the back of the retina, the back of the eye. A fundoscope is a machine that can take a picture of that. So this is a fundoscopic image of an eye, and we have, in fact, if you let me think about that. Have I given you enough anatomy for you to tell me whether it's a right eye or a left eye? Yeah, well, can anybody guess? The fovea, as I told you, is the center of your field of view, right? That's the little depression in the eye where you have the high concentration of photoreceptors. And then the optic disc is where the optic nerve enters the back of the eyeball. Turns out that this is a blind spot. It's a blind spot. And the word for blind spot is scotoma. I should write that on the slide here, I guess. S-C-A-T-O-M-A, scotoma, is a blind spot. But the blind spot is pretty small. So when a neuro-ophthalmologist is coming into your room to look at you, at least the old school ones, I don't know how many old school ones are left, but the old school ones would have a red quilting pin stuck in their lapel. And the reason for this is if you take a red quilting pin, you close one eye and you focus on a certain point, you put that red quilting pin about 23 degrees lateral of the visual axis, you can get to a point where the red dot disappears. It disappears in your peripheral vision. And it's because the light that would be coming from the head of the quilting pin is falling on the blind spot. All right. So the blind spot is medial to the fovea, and because of that, you would guess then that this is a right eye, and you'd be right. But with that pin, everybody has a scotoma, and there's not like a black spot there because your brain takes all the stuff that's around it and kind of smudges it together. It's like higher cortical processing kind of takes all of the peripheral image and sort of fudges it so you don't notice that there's a blind spot there. But sometimes if you have a problem with your optic nerve, maybe there's high intracranial pressure, or there's a million things that can happen to your optic nerve, that scotoma can get bigger, and those neuro-ophthalmologists can actually map out the size of that scotoma really quickly, without having to stick a person on the visual field machines. That's the way that you do it quantitatively, of course. You put a person... Has anyone had one of these visual fields before? Some of you must have. Those with glasses certainly have. It's where they have a little red light and it beeps and you have to click it. You guys ever done that? Anyways, there are ways of mapping the visual field more accurately, but you can get a pretty decent map of the scotoma in just using that technique. All right, so it may not work. Maybe the people in this row it'll work for. But if you close one eye and you look at, with that eye, the X, so how does this work? So if for the left eye, if you look at the dot, the X should disappear. And if you look at the other eye, you look at the cross and the dot should disappear. And if not, you can just move your eyes around until the dot actually will disappear. And it's a way to see that the blind spot is in fact actually there. Yeah, if you just kind of look at something a little off center, you can get it to a point. Is it working for any of you? Yeah. Wow, isn't that fun? Okay, so the optic nerve is amazing for a lot of reasons. I'm not necessarily trying to sell neuro-ophthalmology to you because it's a pretty hard road, but it is incredible. It is incredible. The optic nerve is the only nerve in the body, that can be visualized from outside the body. Standing in this room, I could visualize, if I had an ophthalmoscope, I could visualize your optic nerve and look at that and actually visually inspect that nerve. That's the only non-invasively visualizable nerve. However, to do this, when you're using these ophthalmoscopes, you have to be... there is a little bit of intimacy about this. You need to get right up on top of it. So if you were ever in training at some point and whatever, you see somebody trying to look in the eye, this is how it happens. You look in the right person's eye with your right eye. You look in the left eye with your left eye. But you have to get real close so you can get around and see the back of the retina. This woman seems to be happy about the health care she's receiving, although this guy is not seeing anything. That is an utter waste of both of their times, unfortunately. Why would you look in there? Why would you want to visualize the optic nerve? Well, the optic nerve can have cupping. And so here's an example of a healthy optic nerve on the bottom left-hand side. And I put a whole bunch of different pathologies in the optic nerve. A person who is trained, a neuro-ophthalmologist, to know what they're looking for can diagnose a wide range of things. Everything from multiple sclerosis, an inflammatory problem of the eye, neuromyelitis optica, various infections. Maybe you have systemic septicemia. You can identify that by looking at the optic nerve. Maybe there's cranial arteritis. You're having some kind of vascular problem in the brain. Is the person on drugs? Is there a tumor of some sort in the brain? Has the person been subject to radiation therapy? Et cetera, et cetera, et cetera. For a person who's trained to know what they're looking for, you can tell an enormous amount about not just the ocular physiology, but about the whole systemic physiology in the body by being well trained in knowing what you're seeing when you look at a person's optic disc. You can have progressive swelling, or it's called cupping. And so here's a pretty cupped A and B are both pretty heavily cupped optic nerves due to optic neuritis. And then there are, we do live in a computer age. I've just been selling you on all these old school methods like quilting pins and getting right up there with an ophthalmoscope to know what you're looking at. But computer images can tell you a lot. So this is actually a high definition optical coherence tomography of my eyes. That is the back of my eyes. What was kind of interesting here, and you can see, so you see this like little bit hypodense region up here, and then you have a darker region. This is the vitreous of my eye, which had become detached from the retina, and I'd gone diving, and I started getting those floaters. Has anyone ever had those before? Yeah, those floaters are just remnants of vitreous that go across your visual field. I'm fine. It doesn't really, it's not bad for my eye necessarily. It puts me at a slightly higher risk of retinal detachment, but I'm okay. That's what I get for going diving. All right. I don't know. I was maybe 12, 15 meters. Not that deep. But I didn't have a good face mask on. I had like little goggles. I was free diving and whatever. Okay, let's keep going. All right. So on to photoreception. With the time I have, I'm unlikely to get through this, so I'll have to pick it up next time. But no problem. First, the anatomy of the retina. So I said there were two layers to the retina. There was the pigmented epithelium, and then there was the neural layer. We're going to start from inside the eyeball, where that vitreous is that I just spoke about. And here's light. It's come through the lens, which is somewhere down in the basement, and light has come up through the eye, through the posterior cavity of the eye. And it's going to go through this neural layer. The first thing it goes through, you may think the first thing it hits would be the photoreceptors, but in fact it's not. First thing it hits are what are called ganglion cells. And in fact, it goes through several layers of cells here before it can get to the photoreceptors. It goes through ganglion cells, which are attached to these bipolar cells. And then bipolar cells are actually attached directly to the photoreceptors. But at each of these interfaces, at the first interface, there are amacrine cells, and then at the second interface, there are these horizontal cells, which lend a degree of interconnectivity between adjacent regions of photoreceptors. We'll talk about that in a bit. There are two types of photoreceptors. Most of you are probably aware of that, rods and cones. The rods give you scotopic vision. Scotopic vision is just low light vision. They're very highly sensitive. Only a single photon can trigger a rod. This is supposed black and white vision. This is your black and white vision, right? The rods. But it's not very highly resolved. It doesn't give you sharp, high definition. It just gives you low light sensitivity. So they're highly sensitive to light but they don't give you a very well spatially resolved image. On the other hand, we have cones. Cones are so-called color vision. Because of that, there are different types of cones. Humans have three types. There's a red peak, a green peak, and a blue peak cone. But other animals have more or less, and they're at different colors. Ravens, for example, see in the ultraviolet spectrum. Partially, they also see in the visual spectrum, in the visible light spectrum that we see in. But for example, like a crow, you know how a crow sometimes has that like purplish sheen on it that you can see. Amazingly, to another crow, a crow looks vividly colored, and with some sort of pattern to them, I guess, that we don't see. So crows, to us, they're just these black birds, but to each other, they maybe look like what we would think of as a parrot, I guess. I don't know. But they're able to pick each other out very readily in a field of view because of the photoreceptors they have in the frequency peaks. A little bit of biology. Another thing you'll notice here is, so here's the ganglion cell level, here's the bipolar cell level, and here's the rod and cones. And these little dots you see are the nuclei of these cells. And the significance of that is that there's a lot of signal convergence. A single ganglion cell monitors many bipolar cells, which themselves monitor many more photoreceptors. So there's this like signal convergence that happens from the photoreceptors down. All right. The actual structure of them is there's an outer segment and an inner segment. An inner segment is the part of the cell that has all the goods, all the typical goods that you would imagine a normal cell would have. Like there's a nucleus, endoplasmic reticulum, Golgi apparatus, the mitochondria, et cetera, et cetera. They are neurons, so there's going to be a dendritic plate right here that is going to send out the signal. And then there's the outer segment, and the outer segment between rods and cones is a little bit different. But in either case, they're both just like stacks of membrane. They're just big stacks of membrane. And the exact structure of those is a little bit different in either. However, in these stacks of membrane, there's a special protein called the opsin protein, the opsin molecule. It's a transmembrane protein that when opsin binds to this molecule called retinal, it becomes rhodopsin. We call opsin and retinal in combination rhodopsin. And retinal is what's called a chromophore, which is just a molecule that absorbs light. Retinal is just a slight variant. It's synthesized from vitamin A. Vitamin A just needs to go through one molecular modification to become retinal. That's what we do with vitamin A. So the whole thing about Bugs Bunny, eating your carrots, good eyesight, all that actually is real. Vitamin A is necessary for sight. All right, so, oh man, I got 1 minute. I'm not going to get a chance to talk about all this, but I will start with the dark current. So eyes are mysterious in so many ways. They're mysterious in so many ways. One of the incredible things about the eye is that there is when the photoreceptor is at rest and is not being stimulated by light, when it's not being stimulated by light, it is releasing neurotransmitter. It is firing. Okay? It is releasing neurotransmitter when it's not being stimulated by light. This is called the dark current. It's counterintuitive to the way you would think a normal neuron would be. When you're getting stimulate, when a neuron is stimulated, you think, well, that's when it's going to start releasing its neurotransmitter. But that is not how photoreceptors work. The specific, the players here, and then I'll let you guys out of here. No, I won't. I'll let you out now. It's 1:51 or 9:51. Okay.